Thanks, Steve. Well, polycyclic aromatic hydrocarbons, or PAHs, can be an issue with biochar, and so that's the focus of this study. We made our biochar out of corn stover, that is leaves and stalks left over after harvest. We pyrolyzed under a nitrogen atmosphere and made the char at 3 different temperatures. Our char was relatively clean, PAH-wise. The US EPA has a list of 16 of them that it considers priority pollutants. They're shown here on this slide. This slide is a little old. I don't have the 750-degree char, but the trends are that at the lowest temperature, there's less than 2 milligrams per kilogram total PAHs, and at the most about 0.25 for any one. That's all I'm going to say, other than that this is well under the land application standards for any state. So the PAHs really aren't much of an issue with this corn stover. For the rest of the talk, I'm going to be talking about experiments using pyrene as a probe compound. There's the structure of pyrene up there, and the other thing to say is, if you want to know more about total PAHs, see poster number 2 in the main hall. We did 2 kinds of experiments. One of them was sorption. This is for the 450-degree char. The sorption was linear on a log-log plot, so this is a Freundlich isotherm. The sorption was very strong. The ratio of dissolved pyrene to sorbed in comparable units is roughly 10 to the 6th. And Freundlich isotherms fit data from all of our chars. This puts all of the fresh chars on one graph. 450 and 550 were pretty similar in this concentration range, pretty similar amounts of sorption. They diverged a little down here. The isotherm has a little steeper slope for the 550. The 750-degree char seemed to sorb the pyrene much more strongly. The other kind of experiments we did are what I call bioavailability related experiments. We didn't kill any organisms or measure the uptake, but other people have studied how well PAHs can be extracted from soils and other material by this chemical 2-hydroxypropyl beta cyclodextrin, which from now on I'll just call HPCD. HPCD extractable PAHs are very well correlated in soils and bioavailability experiments. Bioavailability to earthworms or other invertebrates, or biodegradation by microorganisms in soils. There is essentially no correlation with total PAH content and either bioavailability or biodegradability. This is HPCD extractable pyrene in and a 450-degree char. You notice the bottom, the x-axis is added pyrene. That's because there is so little all PAHs, including pyrene, that it was just not detectable in our extracts. We added different amounts, weren't quite sure what to expect, and we just kept adding 3 different levels in the rest of our experiments. So for the most part, 10 to 15% extractability for the 450-degree char. I'll have more stuff, more extractability results in later slides. We also did artificial aging. This is based on a paper that was published after our study got underway that was really very timely for us. It involves moistening char to 40% of its field capacity and incubating for 1 month. The 3 treatments that we chose were freezing and thawing on a daily basis, or incubating at 60 or 110 degrees C. We checked the weights daily and added water as necessary. This slide shows the effects of the artificial aging on sorption to the 750-degree char. Freeze-thaw made it a little more sorptive. This is the same data that was on an earlier slide. What I'm calling fresh char. The freeze-thaw treatment is up here. A little bit higher, that is, for a given dissolved pyrene concentration. The sorbed content is a little higher. In contrast, the heating decreased the amount by a little bit. They're really quite similar for those 2 treatments. And artificial aging also affected the extractability. This is for the 450-degree char. The diamonds are the data that you've already seen. The different aging treatments are pretty well bunched together. I think that's partly reflects the fact that we did a few of these experiments, got better at it, got better reproducibility, but there's still, looks like there's a little bit of a decrease in the HPCD extractability for the aged chars. We went on from 450 to the 550-degree char, and for the fresh char, the extractability is in the same range as for the fresh 450-degree char. And then we went on to the aged chars, and it was like Mother Nature flipped a switch, and all of a sudden the extractability went way down. For this range of pyrene additions, we couldn't detect anything in our extracts. We had to kick everything up by a factor of 10. And even at that, from this point on, the extractability was more like 1 to 3%. And the results, all of the 750-degree ones were in the 1 to 3% range. So, pyrene and by extension, probably, PAHs sorb very strongly to Korn-Stover char, and Freundlich isotherms fit the data very well. One should not extrapolate things like sorption isotherms outside the data, but I think it's pretty safe to say that putting this char in any more moist environment, you're going to have dissolved PAH concentrations much below nanogram per liter, and that's pretty darn low. Only a small fraction is bioavailable. Depends on what temperature the char was produced at. It may be in the 10 to 15% range, or there may be 1 to 3%. Aging has small but significant measurable effects on both sorption and extraction, and by extension probably bioavailability. And I'd like to wrap up just saying a few things about water-soluble black carbon. This has been, if not a hot topic lately, at least a warm one. There's a bit of controversy about whether biochar or any kind of black carbon really is stable in soil because of the water-soluble fraction. We've noticed that when we centrifuge our suspensions before we analyze the supernatant, that there was a bit of color. It looked a lot to me like lake water. And now at the end of the project, we're just starting to look at this a little bit. This is our 450-degree char. We put quite a bit of it, about 1 gram per liter, let it extract for a week. You can see there definitely is a color to that. This dissolved organic carbon concentration is almost 10 milligrams per liter. That's pretty high, and I think that explains the color. The pH was pretty high too, consistent with a lot of the things you heard this morning. Not only does it look like lake water or stream water to the eye, it looks like it to a spectrophotometer too. Here is a UV visible absorbance spectrum of our water extract. An absorbance at 250 nanometers of 0.35 is pretty high. There's a lot of stuff in that water. And for comparison, here's a natural water sample. This is a high TOC groundwater that I recently had in the lab. A lot of the same features in these two spectra. The exponential decrease in absorbance as you go to higher wavelengths, and also there's a little peak superimposed at about 270 nanometers. So this suggests to me anyway that the so-called water-soluble black carbon probably has some structural features in common with natural organic matter. We also looked at it with fluorescence. This is a three-dimensional excitation emission matrix, essentially a combination of lots and lots and lots of fluorescence scans. The excitation wavelength is on the y-axis and the emission on the x-axis. The data are all between these two diagonal lines. The brick red areas on the outside have no data. The only feature of this, the feature that takes up practically the whole matrix, is this large peak that's centered at an excitation of about 340 and an emission about 410. For comparison, here's an excitation emission matrix of that same groundwater that I showed before, really very, very similar. So further evidence that there's probably a lot in common between the natural organic matter, some people call it humic substances, and water-soluble black carbon. We also did an acid-base titration. Here's where the pH started out, about 9.2. We acidified down to pH 4, sparged out the CO2, and then titrated with base. The first few additions made very large changes in the pH. This is the range where you'd expect if there were any carboxyl groups, this is where you would expect them to be neutralized. There don't appear to be a lot of carboxyl groups in this water extract. It's only at the higher pHs where the curve started to bend up. So we suspect that there are a lot more phenolic groups in this stuff than carboxyl groups. This is about all we've had a chance to do. There's a lot of other things we'd like to do. We'll just have to see if we actually get to them. We thank Will Gillespie, one of our recently departed colleagues at the Water Survey, and also a farmer down by Tuscola. We thank him for the corn stover. We thank BK Charma and Wei Jing for making our biochar for us. I personally thank Jill O'Connell for lots of really great assistance with the sorption and extraction experiments, and this was supported by the Sustainable Technology Center.
We have time for a few questions. Go ahead, John.
You notice a color change in the 550?
The question was, did I notice color change in the 550? And I haven't run that yet. I'll tell you what I expect, though. I expect a lot less than in the 450. Actually, I did run my memory is failing me. I'll have to look at my notes. I don't think I've done it yet.
Do you have a hypothesis on the aging process relative to the
The question was, do I have a hypothesis on the aging process, and do you mean the artificial aging that we did?
No. Well, I guess that's your example, but in soil, would that be repeatable?
Clarification on the question is, is this actually the aging process in soils? What is my hypothesis? For the artificial aging, it's most likely some surface oxidation. For aging in soils, there's some of that, but you've also got microbial activity going on, and that might be some oxidation too, or some other sort of transformation. The thing that you've got in soils that we didn't do is fouling, if you will, of the surface with natural organic matter, the soluble organic matter. I'm only lately becoming aware of some papers that I should have been aware of a long time ago, of people finding that natural organic matter does modify the surface of biochar. I think that's a big part of it.
You mentioned like the different temperatures and then aging has an effect on the extractability, reducing. So what would be the underlying mechanism? Why the aging or increased temperature of biochars reducing extractability?
The question was, why would the aging affect the extractability? And I think it goes back to the surface modification, creating or taking away some of these oxygen-containing functional groups. It would probably become a bit more polar, at least at the higher temperatures. Yeah, although I'm really not comfortable hypothesizing on this. That's just a guess.